Hello, welcome to Breakthrough from Science Mission. I'm Sadashiva Pai and going to discuss a couple of breakthrough papers published in the week of August 18th. Let's start. First, we'll discuss a paper published by Chu Mei and colleagues in Science Translational Medicine on the mechanism and control of gastric cancer by nervous system. Why is this paper so important? Gastric cancer is the third leading cause of cancer mortality worldwide, with a 5-year survival rate of less than 25%. Authors show that vagal nerve, which signals to the stomach through muscarinic receptors, contribute to the growth of gastric cancers. Vagotomy can prevent gastric cancer in mice and reduce the recurrence of gastric tumors in human patients. Botox or anticholinergic drugs inhibit vagal nerve signaling and may be used as a safer treatment for gastric cancer. Let's look at the data. Authors demonstrate that INS gas mice has a higher tumor prevalence in lesser curvature than greater curvature of the stomach as shown here. INS gas mice that underwent bilateral vagotomy with pyloroplasty or anterior unilateral vagotomy showed significantly less tumors than sham-operated or pyloroplasty alone. Pathological score of dysplasia also showed better results following VTPP and UVT. Similar results are also seen when number of proliferative cells are counted. Two other mouse models of gastric cancer, namely carcinogen-induced N-nitroso N-methylurea and Helicobacter pylori-infected adenosine triphosphatase interleukin 1b, also showed similar results. Decreased volume density of tumor, pathological score for dysplasia, and number of proliferating cells after anterior Botox injection demonstrates chemical denervation blocks gastric tumor formation. Authors also demonstrate gastric denervation at later stages also inhibit tumor progression. Data is not shown here. Authors found variations in gene expression of Wnt signaling pathway in gastric tumors. Either vagotomy, VTPP, and UVT or Botox treatment induced downregulation of CD44 in gastric mucosa of INS gas mice. Vagotomy also reduced the expression of Wnt target genes in MNU-treated mice. Gene co-expression analysis showed LGR5 stem cells and muscarinic acetylcholine receptor 3 in sorted cells from LGR5 GFP mouse stomach, but other subtypes of muscarine receptors were little expressed in these cells. Also, combination of M3 antagonist and chemotherapy reduced cellular proliferation of the tumors, and M3 knockout mice had less tumor induction and smaller tumor size when compared to wild-type controls. Similar results were also obtained using in vitro cultures, data not shown here. Authors show Wnt signaling, neurotrophin signaling, and axonal guidance pathways were activated in human cancerous tissue. PGP 9.5, a ubiquitin protein hydrolase, and peripherin, a type 3 intermediate filament protein immunostaining, revealed that in humans, neuronal density was correlated with more advanced tumor stages. So this study provides evidence for the role of nervous system in gastric cancer and also a new therapeutic target. Next, let's look into a paper published by Julian and colleagues in Nature Medicine. Authors demonstrate that deregulation of G-alpha-S may contribute to medulloblastoma formation. Why is this paper so important? Genetic alterations driving medulloblastoma initiation and progression remains poorly understood. Authors identify GNAS encoding G protein G-alpha-S as a potent tumor suppressant gene. Ablation of the single GNAS gene in mice is sufficient to induce sonic hedgehog associated medulloblastomas. G-alpha-S suppresses sonic hedgehog signaling by regulating both cyclic AMP-dependent and ciliary trafficking of hedgehog pathway components. Let's look into the data. Authors were able to correlate low GNase expression tightly with significantly decreased overall survival within SH group of tumors, which comprise approximately 30% of all medulloblastomas. Authors developed knockout of GFAP genus mice, and all of them developed medulloblastoma-like tumors at adult stages. And the neoplastic cells were highly proliferative as indicated by extensive expression of Ki67 and was confined to cerebellum. 100% of GFAP genus animals succumbed to their tumor around 3 to 4 months of age. Quantitative RT-PCR confirmed that in knockout mice, expression of SH target genes and pathway components were significantly upregulated. Treatment with NF449, a selective G-alpha-S antagonist, resulted in significant upregulation of SH target genes and decrease of intracellular cyclic AMP levels. Similar result is seen when constitutively activated form of G-alpha-S is overexpressed. Data is not shown here. Also, cyclic AMP rising agent significantly reduced expression of SSH pathway proteins, while inhibition of cyclic AMP dependent pKa with two different small molecule inhibitors significantly increased the expression of SSH target genes. Increasing cyclic AMP levels in knockout mice by rolipram reduced tumor size and proliferation of tumor cells were also substantially reduced. In addition, rolipram treatment resulted in significantly extended lifespan of knockout mice. G-alpha-S was highly enriched at the primary cilium of GNPs, and in the presence of SHH, ciliary localization of alpha-S was diminished. In granule neuron progenitors from knockout mice, at the tip of cilia, cells exhibited strong immunoreactivity for GLI2, a SHH downstream effector, whereas GLI2 was hardly detectable in the wild-type control GNPs. Authors also detected ciliary translocation of SMO 7 transmembrane domain protein in the majority of knockout progenitors, while SMO inhibiting protein PTH1 was mainly detected in control GNPs. The tumor cell proliferation rate was modestly reduced in knockout mice progenitor treated with cyclic AMP inducer rolipram or GDC0449. Combinatorial treatment with both drugs caused a greater inhibition of knockout progenitor proliferation than either drug alone. Authors also demonstrate that various GNAS progenitors and mutant tumors from humans have similar gene expression signature resembling sonic hedgehog medulloblastoma. So the authors totally prove the role of G-alpha-S as a tumor suppressor. Thank you for watching Science Mission.